Hello, everyone. I would like to welcome you to QIAGEN's webinar titled Improved RNA-Seq Data Quality from Low Input and FFPE Samples Using Enzyme-Free Ribosomal RNA Removal Kits, which will be presented by Samuel Rulli, Global Product Director for NGS Products. My name is Kasia Deans and I'm a Global Campaign Manager, and I will be your host and moderator today. If you have questions during the presentation, please submit them into Q&A box and we will answer them at the end of our presentation. Welcome to you, Sam. It's a pleasure to have you here today to discuss this very interesting topic. We are looking forward to your talk.
Thank you, Kasia, for the introduction, and welcome everyone to today's webinar. Today I'm going to focus on trying to improve data quality using a method that we developed here at QIAGEN for removing ribosomal RNA or globin RNA or any unwanted RNA from your sequencing experiments. And the reason why we like to remove unwanted RNA is a couple of different things. The first is if we have a highly expressing RNA like ribosomal RNA, which doesn't provide any biological knowledge to us, removing that can allow us to see lower expressed genes with the same amount of input. And this becomes really critical if we have limiting amounts of sample. So if we have 5,000 cells and we're doing something with them, we definitely want to see those low expressing genes in our experimental data. The other thing is an economic impact. A lot of these unwanted RNAs are represented at very high amounts. So if we think about ribosomal RNA, which can be 80% of a eukaryotic sample or even 95% of a prokaryotic sample, removing that can save a lot on our redistribution and sequencing costs. So there's a lot of different factors that can really play into, and this is the reason why we developed out a new method for removing ribosomal RNA. I need to go through this slide really quick. It is a legal disclaimer that basically says that the QIAGEN products shown here are intended for molecular biology applications. These products are not intended for the diagnosis, prevention, or treatment of disease. For up-to-date licensing information and product-specific disclaimers, feel free to download the kit handbooks. You should be able to find those online. Reach out to your local distributor or reach out to the local sales team or technical support. So today, this is pretty much the agenda that I put together. I'd like to talk a little bit about total RNA and the challenges with what I call complete transcriptome RNA-Seq. And for complete transcriptome, this includes trying to capture all the RNA that's there. So in the case of eukaryotic cells, mRNAs and long non-coding RNAs. In the case of prokaryotic cells, it's all the RNA that are there. And in other situations, it may include RNAs where we don't even know what their biological value is, but we want to capture them in some way. I will talk a little bit about the methods for ribosomal RNA depletion, the things that are commonly used in the lab, and how this is a little bit different than how QIAseq FastSelect works. I'll talk a little bit about performance data around QIAseq FastSelect. It's a product that we launched about 3 years ago. If you go to Google Scholar, you'll find that there's hundreds of publications. There are lots of posters out there. People have been using this in their workflow for years, and it's something that's trusted by a lot of researchers that are doing all sorts of different applications. And then lastly, I'll give you a quick summary of what we talked about today. So really, when we think about RNA sequencing, one of the things that we can really look at is the sample input amount and the sample quality. And around a lot of different samples that we look at, such as single cells or two interacting cells or even organoids and 3D tumors, these really have a low to mid-range input amount based on what we're looking at, but the RNA quality is often pretty high. And when we're working with samples like this, we often are able to go through and pull out full-length mRNA, depending on the methods that we're using. As we look at other types of samples, such as stool samples or wastewater soil samples, things that we find in the environment, which are very dynamic. When we look at formalin-fixed paraffin-embedded samples, where we have a lot of biological knowledge and even sometimes clinical knowledge around these, or when we go through and we look at things that are coming from a biofluid. So if we're thinking of getting extracellular vesicles and going through and extracting RNA, or anything that's a swab or some sort of liquid fluid, like something that comes out of a lung sample. These samples tend to be more challenging, and that's really because of the two factors of either there's low amounts of RNA or the RNA is going to become degraded, either during the fixation process for FFPE or because the RNA is in a very dynamic environment, it's going to get fragmented. Traditionally, when we looked at RNA sequencing and we started with our sample input amounts, we were thinking of starting with nanogram quantities or 1,000 nanograms, a microgram of input. And we had the requirement that the RNA needed to be high quality. That means RIN scores of 8 or better, DV 200 percentages of 80, 90%. And the real reason for that, if you think about it, is because the methods that we had for removing ribosomal RNA were two ways. One is we could, bind a probe to it or hybridize and pull it down, or we would just basically forget about that and we would do mRNA enrichment. And both of those methods really require high quality RNA. As we've gotten into more advanced RNA sequencing, when we're looking at all these other types of samples, we run into one of the two problems of either low input or we're going to have stuff that's pre-fragmented like FFPE. So when we think about these samples and we think about removing ribosomal RNA, we need to develop kits that are going to deal with these challenges. So for FFPE samples, the biggest challenge is that it's going to be fragmented. And you need a method that will work on RNA fragments from 100 bases on up. For biofluids, this is going to be fragmented, and the limit or the amount of RNA that we have in those samples may even be below the limit of detection. However, there is RNA there, and we can build libraries from it if we can get around the ribosomal RNA problem and we have very sensitive kits. And in the cases of tissues, one of the things that affect us is not so much the quality of the RNA, but that can be a factor, but really the workflow. And in tissues, we can have low input amounts if we're manipulating the cells, as you can see in this particular picture. But we want to go through and we want to have a workflow that allows us to go from a couple of, a couple of thousand cells in some cases and get that onto the sequencing machine where we may have thousands of samples. So when we're thinking about all the different methods for RNA sequencing, there's a lot of different methods that come up. And based on the goals of our particular project, we may be interested in looking at gene and transcriptome expression analysis. We may be focusing only on gene analysis, or we may be focusing on a targeted set of genes. And each of these methods or each of these different types of data points can be addressed with different types of RNA-Seq library construction. And in the case of the different library construction methods, we would use different number of reads per sample. For today, I'm going to discuss working on a complete transcriptome analysis. And so in these sorts of analysis, we're going through and we're developing out library preparation methods that are the best solution for any sort of fusion gene discovery. This allows us to look at both mRNAs and long non-coding RNAs. We're going to collect the information from all of the expressed exomes. So we're going to be able to look at isoform and exon usage and changes across these. And in this particular type of library preparation, it allows us also to do really advanced things like host pathogen detection or dual RNA-Seq. And this will allow us to look at RNAs that maybe aren't polyadenylated or aren't the best solution for 3' sequencing, especially the microbial content that may be there. And just to kind of orientate you to the major steps in a complete transcriptome experiment, we're going to go through and we're going to isolate the total RNA. From there, we're going to have a ribosomal RNA removal step. We'll go through and we'll do cDNA synthesis, and then we're on to our NGS library preparation once we have the cDNA. The ribosomal RNA step is the one we want to really focus on today. And when we look at a typical eukaryotic cell, shown here with a circle, about 80% of that total RNA that we've isolated is going to be ribosomal RNA. With this ribosomal RNA, if we don't remove it, I can guarantee you that most of your library is going to be ribosomal RNA, and the only way around that is really to sequence a lot deeper, and that's basically going to gobble up any sequencing budget that you have. In addition, if we want to look at long non-coding RNAs, there's really no way that we can do an mRNA enrichment in order to get around this situation. When we look at other interesting things, so here's an example of looking across a lot of different plant species. You can see here that we have basically the same problem. So in Arabidopsis, about 96% of the total RNA is going to be ribosomal RNA. And the same as you look across corn, rice, soybeans, cotton, and even in yeast genomes, or yeast transcriptome data. So if you're looking at things other than human, mouse, and rat cells, this is a big problem. The traditional way of going through and removing ribosomal RNA, on the left-hand side, I have what I call kind of the first generation of ribosomal RNA removal. These were hybrid capture methods. On the right-hand side over here, I call it the RNA-Seq method. This is what a lot of the newer strategies are using. Both of these methodologies are really pre-treatments, where you take your RNA, you go through and you do something with it, you manipulate it, and then you are going to use that as a way to build your, do your fragmentation and then do your cDNA synthesis. So in the cases of both of these, there's usually about 2 hours that are spent, and most of them have over 30 different steps involved. And when you start to think about reproducibility, every time you introduce another step into your workflow, that's a place where you can have differences between your samples. And it's not uncommon to go through and see batching effects based on who's doing the hybrid capture experiment, or batching effects on the RNA-Seq method based on who's going through and doing the setup of it and removing all the different components of it. In the hybrid capture method, this often involves over 30 steps, which include the bead washing step, hybridization of the probes to the ribosomal RNA or the targets, pulling down the probes, and then an RNA purification step. In the RNA-Seq method, this often involves binding the single-strand DNA probes to the ribosomal RNA. In this case, RNA-Seq will degrade any RNA that is an RNA-DNA hybrid. Then you have to go through and remove the DNA probes, and then you have another RNA purification step. So you basically purified RNA at least 2 times going into your RNA library preparation method. The method that we developed here at QIAGEN, we call it the FastSelect method. And the FastSelect method is basically a one-step method. And you add FastSelect to your ribosome or to your total RNA sample. You go through your library construction steps, so it doesn't involve a pre-treatment. And the way FastSelect works is it inhibits the cDNA synthesis of its target. So here you can see in the middle panel here, we're comparing the number of workflow steps for FastSelect versus a couple of different other kits. And on the right-hand step here, you can look at the total amount of time. So with FastSelect, it actually works as you're going through and doing your fragmentation. Our current FastSelect formulation is about a 14-minute addition to the fragmentation step. With this time, as well as workflow savings, it becomes a very universal product that you can use in almost any sort of RNA library preparation method. And so if you think about the steps around the FastSelect method, what we basically sell is a solution. With this solution, you add 1 microliter to your RNA that you're starting with. We typically use 1 microliter if we have a microgram or more input. If you're starting with less RNA, we often dilute FastSelect. FastSelect will work with your standard protocol. So as you go through, the first step that you do typically in RNA-Seq analysis is to go through and do a fragmentation. With that fragmentation, you have magnesium ions in your buffer. You warm up the RNA up to 70 to 95 degrees, and basically you get spontaneously random fragmentation of the RNA. As you cool down that sample, this is where FastSelect will bind to its target. So it's a target-specific product that will bind specifically to whatever we design it to. In the case of ribosomal RNA, it binds to the mitochondria and cytoplasmic ribosomal RNA. In the case of our plant samples, we also target any chloroplast ribosomal RNA. In the case of the globin, it binds to the globin that we designed it for. And our particular globin design is for both adult and fetus or fetal or young child globin isoforms there. After you go through and FastSelect binds, it is bound irreversibly to its target. And then you take the RNA and you use it in your reverse transcription reaction. And basically, we are blocking cDNA synthesis from where FastSelect bound. So there's no need to go through and to add any additional steps. When we're thinking about the RNA inputs that we have, there's really no change to our protocol, whether we're using high-quality RNA or low-quality RNA. So the only difference that we have is around the fragmentation step. So in the case of high-quality RNA, we combine FastSelect with our sample. It takes about 1 pipette step, about 10 seconds. We go through and we perform the fragmentation, which is 95 degrees, typically for 3 to 15 minutes, based on the input amounts and how small you want your fragments. Then we do a stepwise cool down from about 75 degrees down to 25 degrees. This takes about 14 minutes. And then lastly, we take that treated sample and we do our cDNA synthesis and the remaining library preparation steps. In the case of when we're starting with low quality RNA, the first step is the same. We often go through and skip the fragmentation if the RNA is already pre-fragmented. What we will do is we'll warm the sample up to 75 degrees to relax secondary structure. And then as we cool it down, FastSelect binds to the target, and then we go on to cDNA synthesis. So it's basically one workflow with a little bit different timing based on the RNA quality that you're starting with. We've generated a lot of data that you can find on our website. We've looked at all sorts of good high quality samples. We've looked at low quality samples. This is just an example of starting with a standardized FFPE type of sample. So this is the Horizon FFPE RNA reference standards that are used for a lot of the fusion gene product development here at QIAGEN. And what I'm showing you here is the results of with and without FastSelect. So in the left-hand panel here, we have pre-treatment using the RNA multiplex positive control sample. Without any FastSelect, we see about 72% of the total RNA is ribosomal RNA. After using FastSelect, we get this down to about 2.5%. On the right-hand side here, we have another FFPE sample. It's the negative control sample. Without FastSelect, we see about 74% ribosomal RNA. After FastSelect, we see it down to 3.5%. This could be a little bit further optimized to remove more ribosomal RNA by adding a little bit more FastSelect or working a little bit with it. For most researchers, this is greater than 95% of the ribosomal RNA has been removed. And with this standard treatment, following the standard protocol, it's more than adequate to put on a sequencing machine. We've also looked at whether FastSelect will affect the gene expression components. We've done a lot of experiments with different amounts of input. So this is just an example of starting with 1 microgram or 100 nanograms of universal human reference RNA. It's basically a whole bunch of human organs that have been ground up and the RNA has been extracted. We used FastSelect HMR, which is our human mouse rat ribosomal RNA product. We used a QIAGEN library preparation kit, went through and sequenced this on a NextSeq using a 2x75 sequencing, and then we did our mapping with a commonly used tool here at CLC Genomics Workbench. Without going through and using ribosomal depletion, we had the amount of RNA that we used is 1 microgram and 100 nanograms. After we went through and we used FastSelect, we got this down to only 1 to 2% of ribosomal RNA remaining. And then we looked at the correlation between the input amounts for the gene expression. And 100 nanograms versus 1 microgram, you can see a nice linear correlation here, meaning that we haven't corrupted the gene expression component here. Looking down here at the amount of exon, intron, and intergenic reads, you can see that the 100 nanograms versus 1 microgram is that we don't see any change in the distribution of the exon, intronic, and intergenic region here. So in conclusion, FastSelect effectively removes the ribosomal RNA. You don't see any change in the read mapping or the gene expression components. Here's an example of using globin. Here we started with 100 nanograms of human whole blood total RNA. We used the globin product with a QIAGEN RNA library prep kit. And without FastSelect, we had about 88% of our reads mapped to ribosomal RNA. After going through and using the ribosomal RNA and globin kit here, we have about 1% left. With globin, we had about 82% mapping to globin, and after FastSelect, it was down to less than a percentage left. And you can see up here with the treatment versus no treatment, with the human no treatment, you can see that we had only a small percentage of our reads mapping to the exon, with most of it mapping to an intron, which is basically the ribosomal RNA. After treatment, you can see now we have about 40% of the reads mapping to exon, some of it to intron, and then the rest of it to the intergenic region. We've also developed out a kit for pan-bacteria applications, and this is the kit that we use when we're starting with some sort of sample where we know there's bacteria present, or when we're going through and looking at dual RNA-Seq. For FastSelect, we call this our 5S, 16S, and 23S, so that we can remember that we've designed FastSelect against the 5S, 16S, and 23S ribosomal sequences. The way that we did this is basically community-level blocking of these bacteria or pan-bacteria product. We designed this against the SILVA database for the 16S and the 23S sequences. There were about 600,000 unique entries for 16S and about 170,000 entries for 23S. And then we also used a 5S database that had about 7,200 unique entries. We went through and we demultiplexed or aligned everything, got to the unique regions that were in there, and basically tiled FastSelect across these regions. We've tested this with anything from 5 nanograms to 1 microgram of total RNA input, and we've used a lot of different RNA library kits here, including the NEB Ultra 2 Directional, the TruSeq Stranded Kit, and a few others that we now have up on our website here. Just to show you some data here is we're looking here first at an E. coli. This is DH5 alpha E. coli total RNA that we acquired from Thermo Fisher, 1 microgram and 100 nanograms. We have data here of no depletion and FastSelect depleted, and then going through and using this with a QIAGEN NGS kit here. On the upper right-hand corner here, you can see the percentage of reads that map to E. coli after going through and using FastSelect. So down here on the lower panel here, without treatment, we had about 98% of the total RNA was mapping to ribosomal RNA. After going through and using FastSelect, you can see it gets down to about 1 to 2%. And this is with the E. coli K12 strain. In the lower left-hand corner here, we're looking at the gene expression components here. You can see a high correlation regardless of the amount of input that we're looking at. And then we went from a single E. coli species up to something that's more complicated. So here we're looking at a couple of different panels of different bacteria. And we have here a whole, what's called the 20 strain whole cell material from ATCC, the skin microbiome collection, and the oral microbiome collection. Each of these have a series of different commonly found microbes that are in those different areas here. Same exact experiment. And here you can see that we start with, on this particular sample, we had about 95% before we treated with FastSelect, and we get down to about 15%. As you go through here on the bottom data, what we've mapped out is the percentage of ribosomal RNAs in all of that and the individual species that we have in here. So you can see here without treatment in blue, and then with the FastSelect in red. And you can see there are a few species in here where FastSelect, you can see some residual ribosomal RNA. And this is basically comes back to what data we have in the database we can design for, what data we don't know about, we basically don't know. And so there's not a design for that. And that's why we see some of this ribosomal RNA sneaking through. To combat this problem, what we have is the ability to make custom panels. So after we go through and do the sequencing, often we can see that there may be 15% left in a particular type of sample. And then we can take that ribosomal RNA, put it into our design algorithm, and add back and build a custom FastSelect against those specific targets and basically use it as a booster panel. So for a lot of our experiments that are outside of well-annotated samples that are single species like human, mouse, and rat, or just E. coli, after we've done that first experiment, we're able to customize that booster panel, and we can remove whatever residual FastSelect we see in those samples. The other thing about FastSelect is I've shown you individual FastSelects, but they can be combined together. And so we do have some researchers that go through and they typically do dual RNA-Seq, and they add the human and the bacteria together, 1 microliter of each, follow the standard protocol, and now you have an additional benefit of getting rid of any human ribosomal RNA and any bacterial ribosomal For samples that you find in the environment, we can combine bacteria, plant, and yeast together, and we can have something that if you're looking at the microbacteria that grow around particular plants or on plants, we can use that as a particular thing. Here's some other examples here of looking through different bacteria stuff. So you can see we're now comparing supplier TF, one of their products or a couple of their products versus FastSelect. And you can go through the data here. And the short of the story is that the FastSelect outperforms a lot of the existing kits in the market for bacteria. And there's a couple of different reasons for that. One is that FastSelect is a newer product, so it was designed against more RNA-Seq data that we had at the time. And that allows us to go through and to pretty much have a more broad pan-bacteria product. The other thing is that the product is designed to work on fragmented RNA. And if you have any fragmented RNA in your sample, a lot of the existing hybrid capture or even some of the RNase H methods will fail because the density of the probes or the probes just basically can't identify their target when you have highly fragmented RNA. And lastly, here's an example of a gut microbiome that we've done with the FastSelect product. In this particular experiment here, you can see the comparison of a couple of different samples. Supplier TF, their product ME, supplier TF, product RM, versus FastSelect, where we see a large ribosomal RNA removal. A lot of times when you see a product that doesn't perform as well, it's not that the product doesn't perform well, it's just that whatever is in that sample, there's often a few regions that get through, and those few regions that get through may have a lot of reads on it. So that's usually what we see the problem goes. On a situation with plants, plants are usually very heavy in ribosomal RNA. This comes from both the cytoplasmic mitochondria and the chloroplast. Here I'm showing you examples of all the individual plants that we've tested with our plant product. And you can see the amount of ribosomal RNA goes from a large 96% down to a very, very residual 1 to 10%, depending on the individual species that you're looking at. The plant product was also designed as a pan-plant product. We know it works on most of the commonly used model species that we have in the laboratory, a lot of the commercial crops out there, and then we've also built some custom FastSelect to look at particular things really around some algae situations and around citrus fruit currently. And if we look at the impact that we have on the number of reads, where we can really think about this, in this particular situation here, we're looking with and without FastSelect and the number of reads that map to the genes. And this is basically what you're paying for when you do the RNA-Seq stuff. So you can see here with the Arabidopsis, we have without FastSelect very, very little mapping to the reads. After FastSelect, you can see we have over 50,000 of these reads mapping to genes out of about 60,000 total reads that we use. And you can see the same situation for everything. So if you want to improve the amount of data that you get from a sample, FastSelect is definitely one of the methods to use. And just a percentage-wise on the number of genes that are detected. In the case of Arabidopsis, we're able to increase the gene content 18%. In the case of all these different types of commercial crops, we're able to increase the number of genes detected by at least 30 to 60%. And again, we have a lot of data available on the website. If you're interested in looking at FastSelect and how well it works, I'd go to the website and you can see we usually update whatever information we get from ourselves, so our scientists and customers. So we have a mapping of no FastSelect, with FastSelect, the percent left, and the percentage of ribosomal RNA removed, which is basically the difference of this divided by how much you started with. In our handbooks, we also have a bunch of different tables. Our human, mouse, and rat, I can see some questions about, does human, mouse, and rat cover other things in human, mouse, and rat? And we've tried it on cow, pig, horses so far, and there's probably about a dozen other things that researchers have tried and published at this point. And in general, everything as far as the efficacy of FastSelect will really be based on how much homology you have between human, mouse, and rat ribosomal RNA. And so typically we see a good 80 to 90% removal. If it's necessary where we miss some regions, we can come back with a small custom FastSelect booster panel that we can spike into the sample to make it work. And lastly, some data around yeast. We can see with yeast that we go from 94% ribosomal RNA and down to removing basically all the ribosomal RNA. Sometimes around some of the data that you see here in red, you can see the unmapped percentages actually increases. And this isn't really bad data. What we often find is that when we're looking at the performance of FastSelect, one of the things to keep in mind is that we are mapping this back to some sort of reference type of thing. And for a lot of the references that are used within the pipelines that you may be looking at, a lot of it only contains the mRNA data. And so as you start to use FastSelect in your experiments, if you get a lot of unmapped reads, the first thing I usually do is go to our bioinformatics scientists and ask them, what are you using as a reference gene set? And for a long time, we were only using mRNAs because we were doing either mRNA enrichment or we didn't really have the power to see the low expressing RNAs that are there. And one of the things that we always stress in our handbooks is that if you see a lot of unmapped reads, make sure that you go back and you can actually map those or change the reference sets that you're using for your pipelines. In the case of this yeast experiment here, I know that these 19% that is actually unmapped, these are mapping back to non-coding RNAs and other RNAs that are being expressed within the sample. And some of it's just random noise that you find, but they are a lot of low expressing things that you can map back to the genome or to other regions that don't fall within a traditional open reading frame, but maybe do have some biological value. So as the different species that we've brought online, we have human, mouse, and rat, a pan bacteria. We have one where we combine this together. We call it the epidemiology panel. We have one for fly, which was designed around Drosophila and works for things that are related. C. elegans, a worm. The fish was designed around zebrafish, rainbow trout, and a few other types of fish that are commonly used. Some of these have to do with salmon and other food fish. Plants, yeast, and then, of course, custom that we can do. FastSelect is compatible with the workflow that you're using in your lab. I showed you data using the QIAseq stranded kit because that's the kit that we often have here at QIAGEN on the shelf. But we've also tested it with the TrueSeq workflows, NEB, Kappa, Takara, the QIAseq UPXome RNA library kits, which is a new kit we have coming out. We've used it in single cell applications with different types of methods. So really, it's a universal type of tool that you can put into almost any sort of RNA-Seq workflow, as long as the RNA-Seq workflow has a cDNA synthesis step. We've used it exclusively on short read sequencing because that's what mostly that we're doing. However, customers have also used it on long read sequencing. And now there's at least 6 publications that come to mind where researchers have used FastSelect with an Oxford Nanopore device. They basically run the same protocol that I showed you, except they tried to limit the RNA fragmentation. So they warm the sample up to about 75 degrees, just long enough to relax secondary structure, have the FastSelect in the solution, cool it down, and then they make cDNA from that. We have some researchers that are trying to use it on direct RNA sequencing on an Oxford Nanopore. I would say that it hasn't been 100% successful, but we can see removal of ribosomal RNA. And what we think is it basically becomes too big of a molecule to fit through the pores to be sequenced. One of the things that we've noticed when using FastSelect is that the amount of RNA in the sample is dramatically different than using other ways of ribosomal RNA removal. When we've tried to use hybrid capture systems, the first generation of ribosomal RNA removal, where we're using biotinylated probes and pulling out ribosomal RNA, we always had excess ribosomal RNA in our sample. When we've used RNase-H methods, we find that they're more effective than the biotinylated probe capture stuff, but sometimes we still have ribosomal RNA in our sample. One of the things to keep in mind is that the RNA library construction that you choose is often based on the amount of RNA or cDNA at that point that you have in your sample, and that you may need to dilute the adapters more or add a couple of more cycles to the amplification step because we find we're removing a lot more of the ribosomal RNA, or in the case of globin, it's completely disappeared. We've recently redesigned and built a new library kit, which we call the QIAseq UPXome RNA library kit, which allows us to do a couple of unique things with it. First, we can start with about 500 picograms, but it has a broad range to go up to 100 nanograms of sample. The way that we have put together the kit is you can choose to do complete transcriptome or 3' RNA sequencing. And this allows you to choose the right protocol for your application. In the case of 3' RNA-Seq, we often still include FastSelect in our samples because there are some tracks within ribosomal RNA. And even if you're using an oligo DT to do your priming, you can pick up anywhere from 5 to 30% ribosomal RNA just with an oligo DT. One of the things around the FastSelect is it allows us to do some cDNA pooling. So we can take the cDNA and pull it together based on adding a sample barcode at the early steps of this. And the sample barcode allows us also to ensure sample security. With this, we include a data analysis feature, and also this is built exclusively for Illumina NGS experiments. With the data analysis feature, we also include this with our FastSelect product as well. And the reason why we have this cloud-based data analysis is this is a way that if you're working in the lab and you don't have access to a bioinformatics scientist for all the experiments that you do, it allows you to quickly go through and make sure that FastSelect is working the right way. So I'll talk a little bit more about the data analysis feature at the end, but when you buy QIAseq FastSelect or any of the RNA library kits at QIAGEN, there is a quick and easy way, if you can upload FastQ files to the cloud, for you to go through and to look at how well FastSelect's working, and it will also do other things like calculate differential expression and pathway analysis and things as well. For our new RNA library kits, these were built specifically around FastSelect in mind, so they are quick and easy and efficient library construction kits. The total protocol is done in less than 6 hours with really minimal hands-on time. Most of the hands-on time is based around going through and doing the bead cleanup steps, which if you're lucky enough to have a robot in your lab, you can actually automate this. So with the FastSelect and the RNA fragmentation, this takes about 20 minutes to kind of set up in your lab. You get the things out in the morning, get a cup of coffee, check your emails, and about 40 minutes of total time. It's about 20 minutes to do the fragmentation and everything in there. For cDNA synthesis, we use a template switch method here, which we find is a lot more sensitive than some of the other methods. For cDNA synthesis, you choose which primers you want to use in the kit, whether it's a oligo DT, a randomer, or mix them together for complete transcriptome coverage. We use about 30 minutes to set up all the reactions, and we use about 90 minutes to go through and to do this cDNA synthesis. So a lot of times to play on the internet, whatever you want to do. At this step, our kits have an optional step, which is cDNA pooling. Each of the samples get a unique sample ID at this point. And the idea around the unique sample ID is that this gives us sample security. So we don't have to worry if somebody mixes a plate up or it turns backwards. We often have many people touching the same samples. It also allows you to pool things together, which for certain applications, this might be an advantage. So for like high throughput applications, or when you're going through and you want to cut down on plastics. By cDNA pooling, we can save up to 95% of the downstream master mix beads and plastics that are used within a workflow. Then we get into our first bead cleanup step, which takes about 60 minutes to do if you do it manually and take your time. And then lastly, we do an amplification step and the indexing step. So we PCR on the sample indexes, we use unique dual indexes, and the amount of total time is really based on how much sample you have there. We just change the amount of cycling time that we do. One more bead cleanup step, and then you have an optimized library with FastSelect for low input applications. And this is just an idea around the cDNA pooling. If you've run into situations where you want to have higher throughput without going through and adding automation to your lab, or you're just interested in saving plastics and downstream reagents. As we go through and do this cDNA pooling, we're able to incorporate in the FastSelect ribosomal RNA removal. During cDNA, everything gets a unique barcode, and then all these samples can go into a library. Typically, we pull anywhere from 8 to 24 samples together based on the volume that we have there. They fall into a nice Eppendorf tube. And again, this will save on the downstream master mix beads and the plastics that you use. And this also gives you a greater efficiency as far as the price that you can pay for one of these library preps. For the last couple of minutes here, I'd like to talk a little bit about data analysis, because we do include data analysis with FastSelect in our UPXM RNA library kits. And again, one of the reasons why is that as you're going through and putting together protocols in your lab, you want to make sure that if you're adding FastSelect to your NEB, Illumina, or Kappa, or Takara library prep kits, that it's actually working. And for a lot of the bioinformatics that are out there, when you turn your FASTQ file over to a bioinformatics scientist, they don't really want to map ribosomal RNA. That often ends up in the reads that don't map in, and you don't really know what's going on there. So this is really a quick and efficient way to see what ribosomal RNA is there, how much you're removing, and then it also is a full-blown RNA-Seq analysis pipeline if you don't have access to one. And within this pipeline, this is kind of showing you a dashboard here from a project I was working on. This is a demo project. If you log into the RNA-Seq analysis portal, it will pre-populate a bunch of data in there. And there's actually a lot of the FastSelect data that I talked about has been loaded into the website. In here is a demo project here, and I have 5 different experiments. The way that the RNA-Seq analysis portal works is that when you buy one of the products from QIAGEN, there's a lot number on the box, or we also ship them with some cards, and those have a unique code on it. When you go to the portal, you put the code in there, and this allows you to go through and have a credit. And we charge a credit anytime we do the alignment to the reference genome. After you do the alignment, you can go through and take those normalized gene expression values and compare them to whatever you want as many times as you want. And the idea here is that when we do the alignment, that's when we turn on the processors that we have sitting up in the cloud. That takes a little bit of energy and time. And then after that, when you have that flat file, which has the genes and the gene counts, then you can go through, it's a very small file, but you can use it as your experiments grow. So if you do 3 experiments the first time, that would be 3 credits. If you come back and you do another 3 experiments, we would charge you 3 credits. But now you have 6 samples you can put together. And so that data allows you to grow allows you to grow your experiments and your analysis as you put more and more data together. There's a couple of different tiles here. One tile here is the experiment summary and QC. This is where we can go through and look at the quality of the sequencing and how well FastSelect is working in the samples, as well as the genes that are found and everything around there. There's another tile in here that shows differential expression. And then the last tile down here is basically a Venn diagram where we can compare the genes across many different samples and look at what genes are unique to individual samples and which ones are common. Just a quick, another thing I'd like to point out here is that these products, these individual projects can be shared. So if there's one person in a lab that's working with a particular data set, you can share that back to any of your colleagues. And so if you're working in the lab and your PI or somebody else on your team or in a different institute wants to look at the data, you can click on Share Projects, enter their email address, and then they will get sent an email saying there's a project available for them. They can simply log in and they will have access to your folder to go through and look at the data that you're sharing with them. And they can also go through and change some of the thresholds if they wanted to look at certain gene expression characteristics. This slide kind of shows you the differential gene expression dashboard here. After we've gone through and we can see that FastSelect is working, the next thing to look at is, well, how's the gene expression doing? And often what we want to compare is with and without FastSelect. We want to normalize the reads a little bit here to make sure we get the same kind of coverage there. But this is a great tool also to go through and prove to yourself that the gene expression component doesn't change when you add FastSelect. And what I'm showing you here on this dashboard in the upper left-hand corner is the genes and their full change values, and also the different statistics that we use. We use both the false discovery rate or the corrected p-value, and then also the uncorrected p-value here. I often use the FDR p-value, but there are some instances where you want to use the student's t-test p-value. In the middle here is a volcano plot. So here you can see to the right are genes that are upregulated. To the left here in gold are genes that are downregulated. And I've kind of filtered this data set a little bit here. So the ones that are in gray are outside of the bands. And the bands are set down here. You can either drag this, or I have the amount upregulated and a p-value here. Upper right-hand corner here is a heat map. And this is a dynamic heat map. As you drag and drop and move these lines around and select which genes that are here, basically this heat map will kind of move, and it allows you to look at the clustering of the samples as well as the clustering of the genes. And you can visually actually pick out biomarkers from this. You can see genes that are up and down regulated across the samples, and then as your samples cluster together better, you can start to see that, yeah, these are genes that actually I could use to distinguish whatever samples I'm looking at. The lower left-hand corner is pretty cool. This is coming from Ingenuity pathway analysis. So if you're not familiar with Ingenuity, Over the last 30 years, there's been scientists reading papers, and basically a lot of the published values, the directionality, genes that interact with other ones in pathways or individually, have been curated in this database here. As you go through and you put the samples together to do the experiment, at the point of where we have the differential expression, we send it off to Ingenuity, and Ingenuity returns the results. And with those Ingenuity results, we have different pathways that are shown based on this gene expression dynamics here. We can also get upstream regulators, such as transcription factors that may be driving the changes. We also blast against microRNAs that can be regulating that gene expression stuff, and then also provide back any other biological data that we have. For human, mouse, and rat, we have a lot of data on microRNAs and gene expression components that we can put in there. And as we bring more and more species up, we can add that data back. And just an example here of the different kind of modules that you have. The reason around this is really to be able to go through and look at whether FastSelect is working, look at the library QC. And then the last thing down here is a little box down here. It says, what's next? If you're going through and you're doing a lot of gene expression stuff in your lab and you're starting with RNA sequencing, and then you want to move over to PCR, when you click the what's next button, that takes you to find the PCR assays that you could actually switch to if you want to use PCR as a complementary way to verify your results. And this is just an example of what the IPA window would look like. If you click on View Details, this IPA window here would pop open here. And you can see here we have the canonical pathways, upstream regulators that are microRNA, and diseases and functions. It just basically takes your gene expression and compares against the database and says, this looks like a particular type of cancer sample or something else. It's really useful if you're starting with a sample and you really don't know what's going on. It can give you some quick insights into what kind of other things that you might want to look at, or maybe it would say that this sample looks like this other sample here, this particular type of disease. So if you look at that disease, you may see common transcription factors or regulatory networks that are going on here. And then just an example of looking at the downstream gene targets and finding verification assays, either using digital PCR or real-time PCR. This is available for both microRNA as well as other types of gene expression experiments. So in today's webinar, I talked a little bit about improving data quality, some of the tools that you can use. The FastSelect RNA removal kits are really designed around both low quality and low input applications, but can be used for your traditional standard RNA-Seq that you're using now. It's available for a lot of different types of species, so human, mouse, and rat, which covers most everything in the barnyard, to say. Other types of things like plants and yeast and bacteria. And FastSelects can be combined together. So you can make one master pool of a couple of FastSelects to use on all of your RNA-Seq applications, and we can customize things. Around customization, it doesn't necessarily have to be a gene that we remove. We've used custom FastSelect to remove small RNAs, microRNAs, piRNAs. We've removed tRNAs from some things. We've removed non-coding RNAs. We've removed contamination that we've seen in certain samples. So there were some projects we worked on with human skin samples that had some bacteria contamination and other things that were coming up. And in those cases, we used the bacteria FastSelect with the human FastSelect and a custom one to get rid of some of the other things that were soaking up the reads. The other things around FastSelect is that these can also be used with any other library prep kit that you're already using. It's basically a drop-in module into your existing workflow that can save you 2 hours and maybe a lot of hassle of what you're doing. We talked a little bit about QIAseq UPXM RNA library kits. These were designed specifically around FastSelect. They ship with FastSelect in the box, so that and the protocols that we have been designed around a lot of the applications where we see low input or low quality samples. These kits are a little bit different than a lot of the kits on the market. They use the template switch method, and we've put a lot of features in here for sample security, cDNA pooling, and other things that allow us to work more agile with some of the types of samples that you see here. I talked a little bit about the RNA-Seq analysis portal that we can use for sample QC, for looking at differential expression. This comes standard with any FastSelect purchase and also is standard with the RNA library kits. This is a way for you to go through and make sure FastSelect is working in your sample. One thing around FastSelect that we've also found, and this kind of probably is obvious now, is that as you work with lower and lower amounts of sample, we can dilute FastSelect more and more. And so you can use it and actually extend the reagent life that you have there. The reason for this is when we first designed FastSelect, we were using microgram quantities of input. So as we've gotten down into hundreds of nanograms and 10 nanograms and into picogram samples here, we find we dilute FastSelect more and more. And the reason for this is that when FastSelect doesn't have a target to bind to, it often will bind to the randomers that we're using for the cDNA synthesis. So by diluting FastSelect down, if it finds the target, it will bind to its target. Without having a lot of FastSelect that isn't bound to the target, it doesn't soak up all the randomers that you need to drive cDNA synthesis. And you can overcome that also by spiking in more randomers in certain cases. And then lastly, I showed you the QIAseq RNA-Seq analysis portal included with the products and a way to go through and actually check that FastSelect's working. And it's available with the QIAGEN kits, and it's also available as a standalone component. So if you're struggling and you need RNA-Seq data back tomorrow, this kit or this cloud-based analysis will return anywhere from a couple sample up to hundreds of samples in about 8 hours of time. Since it's a cloud-based application, as long as you can upload the data into the cloud, we will stand up as many nodes as necessary to crunch the data, do the differential expression, and return the ingenuity pathway analysis data back to you. Typically 4 hours, usually at most 8 hours from the time you start. So I'd like to go through and look at the questions that we have here. I see there are a lot of questions in here, so I'll answer as many as I can in the time that we have remaining. So a couple of different questions we have around here is the, some applications that have to do, for example, with laser capture, micro-dissection, pulling nuclei out of certain samples, something that falls into that kind of low input category. And these are perfect examples of reasons why we've used FastSelect and developed out our special low input kits that we're bringing to market. Really around low input samples, it's really hard to enrich for mRNA because basically you end up losing sample. If you get back 80% of the mRNA starting with nothing, you still have very little amounts that you can build with. In our new UPXome RNA library kit with FastSelect in it, by going through and using FastSelect, we don't need to play around with the sample too much, so we find that we don't lose a lot of sample. And our input ranges are 500 picograms up to 100 nanograms. So if you're able to get a couple of nanograms out of a sample, we don't really care if the sample's fragmented at that point. We would use primers that are randomers as well as oligo-DT to use through the cDNA reaction. And this is something that is a common use for us now in our laboratories. For the UPXome RNA library kits, we expect to have those on the market on August 19th. However, if anyone sees value in that, we do have a demo program and an early acceptor program. So if that's something that can work in your lab, please let us know, and we can talk about getting you those kits as soon as they're available. Good question here about whether FastSelect is compatible with bacterial ribosomal RNA. It is. For all sorts of different applications. Ours is a pan-bacteria product, so we designed it against as many 5S, 16S, and 23S sequences we could find. And then for anything we miss, we often can build a custom panel around that. A couple other questions in here about what FastSelect is compatible with. Is it compatible with the Illumina library preparation, Kappa, NEB? It's compatible with all library preparations that have a cDNA synthesis step. It's also been used with Thermo Fisher's method, where instead of fragmentation, there is an enzymatic fragmentation of the sample. There's a couple of publications out on that, and it works very well. A couple other questions in here about the types of samples that we've tried. We try to keep our website up to date. So if you search for our QIAseq FastSelect on the QIAGEN website, or you can probably just Google that, you'll find that the website, we try to update whatever samples we have. The other place I like to look for is Google Scholar. In Google Scholar, type in QIAseq FastSelect. It's a very unique name, FastSelect, and the reason for that is it does have a very specific match in Google Scholar for people that published it. And you can find almost any sort of application it's been used for, and you can look through and see if someone's tried it on the samples that you're trying, and they've probably published data or a use case around it. A couple good questions in here about the shelf life of FastSelect. Since it's enzyme-free, it's basically a highly modified oligo that we use. The shelf life that we currently have is 18 months. However, we've only had it on the market and been doing real-world testing for 18 months. Our accelerated testing, where we take it and we cook it and we try to simulate a shelf life of longer times, was probably out to about 5 years now. So the shelf life of this FastSelect product is very long. And what we find is that researchers that buy it, they often look at the number of samples they have throughout the entire year, and they will buy it once or twice or whatever. It's available in a lot of different sizes. There's an 8 sample size, a 24, a 96, and a 384 sample size for most of the species that I showed today. So there's, for small projects to large projects. Yep. A couple other questions in here about single-cell RNA-Seq. So at QIAGEN, we've used this with the QIAseq single-cell RNA library preparation kits. We've just put that data into the most recent handbook. We use it at what we would call 0.1x or 0.01x, where we're diluting it down 1 to 10, 1 to 100, or 1 to 1,000. I believe about a 1 to 100 dilution or more works exceedingly well with trying to get a full transcriptome out of single cells with the method we're using. People have tried to use it with DropSeq and other things. The one thing is that you have to put FastSelect into the reagents that are putting in there. But since it's such a high dilution, it's something that some researchers have tried. I don't know if they've been able to optimize it. Any sort of plate-seq method, though, works exceedingly well with FastSelect. A couple of different questions here about low input, about the efficiency, and what does it happens with the RNA-seq data when you have low input stuff. Around low input applications, when you look at the data, we often have a cutoff of a certain number of counts per gene that we use in a lot of the data we put on the website. That's allows us to get rid of some of the stoichiometric effects you may see with low amounts of genes that are expressed in every sample or only in a few of the cells, depending on what you're looking at there. So in the case of some of the samples here, as you get lower and lower amounts, you will see more variability and more deviations around that. And that's more of a universal thing. Whenever we measure less of something, based on the analytical method we're using, there's a certain amount of precision. In cases where we've pushed FastSelect down to lower amounts, what we find is that a lot of the RNA library kits that we used usually has a cutoff of 20 nanograms of input, or some of them down to 5 nanograms. Depending on the library kit you're using, if you get down below a certain amount, you're going to have more variability. And there's a couple other questions in here about how low can I use, how low can my input be to use FastSelect. That's really determined by the library kits. And that's why we've kind of developed out the low input UPXome library kit that we have, because that allows us to go through and to really make up or to compensate for the additional RNA that we're removing from the sample. A lot of researchers that have low input stuff, when they look at using FastSelect and they remove more of the ribosomal RNA than they've used before, then they often run into problems which are really library kit driven, and they just need to re-optimize some of the conditions that they use, usually additional adaptive dilution and more amplification cycles at the end. A couple of questions in there about how good FastSelect is compared to RiboZero and RiboPools and some other products in here. Compared to RiboZero, the first method and the second methods that come out, the RiboZero is Gold Plus or something like that, and RiboPools, those are RNase-H methods. The RNase-H methods, their limitation is the workflow and the fact that you need to remove the DNA probes and that you're using a hybridization and an enzyme. We find that the enzymatic methods, anytime you're using the enzyme, that can be variable because you can have lot-to-lot variability on the enzyme. And also the workflow is much more complicated than simply adding something to your sample and following whatever your protocol is. You have more variables in there and an extra RNA enrichment step at some point. So what we find around the precision is that the precision is better with FastSelect because it is part of the standard workflow. And then how well the kits work depend on the amount of input you have and how you're capturing that with the library kits and how well the design is. Couple questions in here for RiboSeq. RiboSeq is where you go through, you traditionally digest everything that's not in a ribosome. There are a couple publications out there that have shown using FastSelect for RiboSeq. It does work, but I don't think our tiling is tight enough to remove all of the ribosomal RNA you're going to see with RiboSeq. So if you're interested in a RiboSeq application, FastSelect will work. But one of the things that we're thinking of doing is increasing the tiling density of the FastSelect to make a specific RiboSeq product. If that's something that sounds interesting, type it into the questions box or contact technical support or send an email afterwards, and we can discuss on doing a custom design for RiboSeq stuff. Because we're increasing the number of probes, it would be best to kind of limit that to the number of species or a particular species we're looking at there. One thing I didn't really address too much today with FastSelect is we do have extra bead cleanup steps sometimes when we have too many FastSelect molecules. So in the case of the bacteria product, since it's pan-bacteria, the number of FastSelect probes we're using is a lot more than our human, mouse, and rat product. And with the human, mouse, and rat, we add FastSelect, and then we can initiate cDNA synthesis right after that. And this comes from the fact that we kind of have an idea of what the sample is going to be. We know how much FastSelect we're adding. There's not a lot of residual FastSelect around. But with the bacteria, when you're doing a pan-bacteria screen out of stool samples or soil samples, we really don't know what's there at the beginning. And since we did a pan-bacteria design, we have a lot more probe molecules that are there. And so in that case, the workflow is slightly different. When we go through and we add FastSelect and we cool it down, we do a quick bead cleanup step. And that bead cleanup step is basically to remove any unbound FastSelect that would bind to your randomer that you're going to use for the cDNA synthesis reaction. And this allows you, regardless of what sample type you're working with, when you get to the cDNA step, all of your samples will not have any FastSelect molecules around that can bind to your randomers that you're using to drive cDNA. And we found that this is a much better way to go through and to add that inconvenient bead cleanup step, which actually can be automated. And it allows us to start with the same quality of all samples going to cDNA, and the uniformity is a lot better. I think this is one thing also that when you look at the different methods, if you have a lot of steps in your methods, then that's something that could lead to variability. Some questions in here about the types of indexing that we use on our kits. Most of our kits have gone exclusively to unique dual indexing. For our traditional stranded library prep kits, which are similar to the Illumina, Kappa, and NEB, we have 384 UDIs, which are sold in collections of 96. For the UPXome library kit that I showed you at the end there, our new low input, we have 768 unique dual indexes. We've gone to almost unique dual indexing on everything, and we're phasing out combinatorial indexing because we are often afraid of problems around quality control of indexes, contaminations in the lab, and problems with sequencing instruments. So all the stuff that we do typically sell now and manufacture are around unique dual indexes. The added thing around unique dual indexes is when you look across the different product lines, there are often enough for that particular application to fill a NovaSeq, or even if you use half as many to fill a NovaSeq. Around the UPXome, with that optional kind of cDNA pooling step, this gives you another way that you can combine more samples together. So if we do a 24 sample cDNA pooling with 768 unique dual indexes, that means you could theoretically run over 18,000 samples at one time in a flow cell lane on a NovaSeq sequencing machine. And so if you're looking for a higher throughput method, you can even increase that more by having that cDNA pooling step and internal barcoding methods. Lastly, there's a couple of different questions in here around looking at library construction methods where we're going through and modifying the RNA and looking at that. FastSelect shouldn't interfere with any modification of the RNA. Where FastSelect basically is working is between the fragmentation and that cDNA synthesis step. So if you're modifying RNA by removing a phosphate, adding a phosphate, blocking an end of it for some reason, basically FastSelect won't be bothered by any of that. And that would be perfectly useful or perfectly fine to use with the FastSelect method. So that I'd like to thank everybody for attending today. If anyone wants a copy of the slides, you can type slides to the QA box. We'll get those all to you. And again, I look forward to seeing everybody in the webinar real soon. So thank you and have a good day.
Thank you, Sam, for your presentation, and thank you all for attending our webinar today. At this time, I will now close the session and wish you a great rest of your day. Thank you.